Welcome to the start of a new topic in microbiology, and this is the microbial genetics topic. And this section of the course focuses on what we call the central dogma of biology, because this is really what is at the heart of biology. We have our genetic material, right, DNA, and then we have proteins, which are all the little workers of the cell. And so DNA is important because it tells the cell how to make all the proteins that it needs to function properly. And RNA, as you can see in this picture, is like a middleman. Okay, it is going to help essentially kind of translate the directions of DNA into a language that ribosomes, our protein-making machines, can understand. All right, so we're able to make proteins. So this process of starting with DNA, using it to create RNA, and then using that RNA to build proteins is also called gene expression. And that is because while there's a lot of DNA in a given cell, you can kind of divide DNA into segments. And each segment of that piece of DNA is called a gene. And each gene codes for a specific protein. So one big old strand of DNA might have, and I'm just making this up, might have 100 different genes on it, which are going to code for 100 different proteins. So with gene expression, we will be talking about just a single gene and using the DNA of that gene to create a piece of RNA that then gives the specific directions for its specific protein, okay, that gene codes for. So essentially, it's called gene expression because we are expressing the protein coded for by that gene or that segment of DNA, okay? So that is going to be the big picture, the big ideas that we're going to talk about in this section. So I really divide the microbial genetics conversation into a couple of parts. Like first, we're going to talk about DNA and the features of DNA, and then go into DNA replication, right? Because as a cell divides, that new cell that's made needs a copy of DNA. So we're first going to talk about DNA replication. Then we're going to move on to talk about how DNA is used as a template to create RNA, and that's a process called transcription. And then, hold on, I'm trying to get my mouse in the right spot. There we go. And then that RNA that's made is going to be used to help the cell build the specific protein. And that process is going to be called translation. Okay. Now, once we get through DNA replication, and then we move on to transcription, move on to translation, at that point, you're going to be really familiar with features of DNA, features of RNA, and features of proteins. Then we're going to kind of move on and take a look at mutations and how a mutation, which happens at the DNA level, has this cascading effect, right? Because if the DNA changes, then the RNA that's made from it will change, and therefore the protein may also change, right? It can have like this domino effect. So then we'll talk about types of mutations, and then we'll wind up this section with a conversation about horizontal gene transfer, which is about how bacteria, some bacteria, can acquire DNA from other sources, okay? So those are kind of the 5 big topics that we're going to talk about in this section. So brace yourself, y'all. It's going to be fun.